Okay, so we are on. Okay. Okay, so thank you again for coming here in Padua. So today we will have the first webinar, okay, into the Arqus Verde project. Okay, and yeah, we will talk about what we're doing here at the Botanical Garden, and we agreed about discussing about these three topics that you see here in the slide. Okay, so the first one is related to the definition of the strategies for the management of present and future living collection. Okay, so starting from the beginning, we know that the conservation activities in botanical gardens are not just one activity, but are a lot of activities that are integrated each other. Okay, and we have living collections that are represented by plants that comes, let's say, from our local plants, from local flora, or that could be also plants that comes from other area, and so that are cultivated outside their natural habitat. Okay, and we can have living plants, we can have other part of the plants that are stored, for example, as in germplasm banks. And beyond these conservation activities, there are many other activities related to the functioning of the botanical garden, such as the education. So we have visitors, we have schools that comes here, and we do activities to present what we are doing here and which kind of collections are we preserving here in Padua. Of course, we are devoted also to horticulture activities, so to the development, for example, of protocols for the propagation of plants that could be used then for restoration projects and so on. And let's say that in the very last few years, we are also pushing a lot on the research activities related to the plants that are collected here in the Botanical Garden. So, for example, I was hired 1 year ago in order to also, let's say, fulfill this aim, okay? But let's say that in the last 3 years, the number of groups working here at the Botanical Garden went from 1 to 4 right now, and we are planning to expand also the number of master, PhD, and postdocs working here, okay? So, okay. Botanical gardens are, of course, extremely important for the conservation of plants. We know that more than 20% of the world plant diversity is at risk, and 60% of the threatened plant species are actually cultivated and conserved in botanical gardens. So, on a conservation point of view, the role is extremely important. Okay, but there is a but. Okay, since it is true that in the last, let's say, 50 years, the number of taxa that are now cultivated and preserved in botanical garden has rapidly increased. Okay, so you see here, this is the global data on the global network of botanical garden. So this is true, but let's say that starting from 2008, okay, this increase just stopped. And we're now in a period where we are in the plateau, okay, meaning that we are not able, or we are not increasing the number of taxa that we are preserving in our botanical gardens, okay? When looking not only at the number of taxa, but at the amount of genetic diversity that is preserved in this collection, you see that this plateau has been reached even before than 2008, okay? So you see here in this box here, you see this is in the, should be green color, okay? You see the trend of the phylogenetic diversity over the year, the global phylogenetic diversity concerning botanical garden. You see here that starting from 1990, we are in this plateau, okay? So what is happening is that we reach the peak capacity, okay, for conservation of plants in the botanical garden, meaning that we may not be able right now to increase at infinite level, okay, the number of taxa and of the phylogenetic diversity preserved, okay? And another thing to consider is that we do not only have to consider the number of species, but also the number of individuals per species that are conserved in botanical gardens, since preserving the genetic diversity and yet the diversity of plants means also that we have to conserve all the plasticity, of the plants, for example, for the threatened plants. And again, when considering the network of Botanical Garden as a meta collection, so as a unique collection, you see that this is the trend of the mean number of individual per species in the last year. So it has increased, which is good. But again, we reached the peak, and we are not able anymore also to preserve a higher number of individual per species. And this is also what is happening here at the Botanical Garden of Padua. So we have a limited space, so we cannot think about preserving all the species that we want to preserve. We do not have, for many species, we just have one or two individuals. So that what we want to do right now is to try to understand how to improve the conservation value of our collection. And we have to face these challenges here presented in the slide. So we have a high diversity of plants or collection. We have plants spanning from very arid environments to more tropical ones, and of course the climate in Padua is not suitable for all these plants, and so the management of some taxa could be really painful. And of course, not only in our botanical garden, but one limitation for increasing also the number of taxa is the horticultural experience of the people working in the botanical garden. There are some taxa that requires very specific ecological conditions, and so it might be that not all the people are aware of this or are able to reproduce the exact same ecological condition as in natural environments. And of course, if we look at the distribution of botanical gardens in the globe, we see that most of them are concentrated in the upper part of the globe. So that for species that are threatened and must be preserved and that belongs to the southern part of the world, we do not have dedicated spaces, with already a climate that is suitable for their conservation in the area. So for all these reasons, again, there are limitations, for the conservation activities in the botanical garden. And so we asked what we can do, in order to have a more clever, let's say, management of the already existing living collection in our botanical garden, and also what's for the future. How then can we assess whether a collection could be suitable in the future? So if we want to start a new collection, we need to have some knowledge or some tool in order to understand whether that collection is suitable, for example, considering the new climatic condition that we will have in 50 years here in Padua. And so we decided to adopt a framework that has been presented by the Botanical Garden of Geneva. And they proposed a methodology in order to assess the conservational value of the living collections. And the idea is that we need to consider 5 different criteria that are the ones that you see here in this part of the slide. And for each criteria, we have to provide the score for the specific collection. The score could span from 1 to 5. And the criteria are heritage, conservation, science, education, and cost. So heritage is something related to the cultural value of the collection for the specific botanical garden where the collection is preserved. So making an example, yesterday I showed you the individuals of the Goethe's pal. That is really important for our botanical gardens. This is the oldest plant here. It's almost 500 years old plant. And so on a heritage and cultural point of view, that plant, and so the collection of palm species has a very high, for us, heritage value. So when we will provide a score for that kind of collection, we will get a high score for heritage. Conservation, of course, is something related to how much that collection fulfill conservation purposes. So, for example, if the collection is represented by a family, how many taxa of that family are listed, for example, in Annex 2, or are threatened species in the UCN database. And of course, the higher is the number, the higher could be the score associated to this criteria. For science, again, this is this could be a criteria related, for example, by looking at the research activities of the research group working at the Botanical Garden. Is the collection suitable or strategic for fulfilling the research activities, or considering the let's say the whole panorama in the literature? Is that collection important on a scientific point of view? Then we have education. Of course, there are some collections that are more used for educational activities with visitors or with schools, for example. And according to the usage, we can then assess the value of the collection. And then we have the costs. Okay. For the cost, we can consider, for example, the number of people that we need for the management of that collection, the number of hours per day or per week needed for the management, and of course the money, the cost associated for buying stuff needed for the collection. Okay, and the idea is to assess the conservational value of collection not only on a qualitative point of view, but on a quantitative point of view. And the idea is that then to have all these scores and resume this score in an index that should be a proxy of the conservational value of that specific collection for that specific botanical garden that is doing this kind of assessment. Okay. And so we think that this could be a useful framework in order to understand which collection we have to focus more, that is already present in our botanical garden, so where we should put more effort for the conservation of that collection or for the improvement of the collection, or when thinking of future collection, we might decide which is the best group of species to introduce in the botanical garden according to these criteria here. Okay. This is an example provided by the Botanical of Geneva. They created this document. It is free to download from anyone. And this is, for example, the assessment of the Auricassi collection that they have in the Botanical Garden. And they provided this resume of the collection, where you have, for example, a brief description, the history of the collection, why the collection is present in the Botanical Garden, who was the first that introduced the collection in the Botanical Garden, numbers related to, for example, the number of taxa that the collection hosts, and then for each of the 5 criteria, a brief description, okay, of the comments, let's say, for each criteria that should justify the score that has been then associated to that specific collection. Okay, so yeah, what we foresee to do, we do not have already implemented this framework for our collections, but starting with Benedetta, okay, we will start and scan all the collections that we have here and trying to provide a conservational value based on these 5 criteria. Okay, so we are taking the methodology proposed by the Botanical Garden of Geneva. Of course, there are some let's say sub-criteria for the criteria that I've listed that might be more suitable for one botanical garden and less for another one. But yeah, we think that this could be a way to standardize how we value our collections. And we think that by sharing this methodology and sharing the value of our collection, we can then improve, okay, the mission of the botanical gardens regarding the conservation of species. Okay. Then moving towards the second topic, as I mentioned before, in the last few years, people here at Orto, the direction, the prefect, pushed a lot on the research activity in the botanical garden. So there is no, okay, admit. We have people that is joining. Okay, yeah. Yeah, should be here. Yeah. There is a lot of energy have been inputted in order to improve improve the research activities here at the Botanical Garden. As you see, we have very large spaces for laboratory and offices, and the idea is to to exploit these spaces at their best. Okay, so regarding this, okay, regarding this, we developed a strategic research plan that is starting this year and will last for 3 years, where we listed all the topics and all the research questions that we want to address in these 3 years. And this strategic plan is based on these 4 pillars, okay? Biodiversity, resilience, historical collection, and conservation, okay? So, for example, we have groups that works with molecular data by analyzing genomes, for example. We will see later an example of the sequencing of the Goethe's palm, the first sequencing ever. And we have groups working on microbiomes, and so how to study the microbiome diversity. Then, of course, we are living in a world of climate changing and global changing in general. And so the second pillar for our plan is to study the resilience of plants towards these changes, okay? And both using a molecular approach. Sorry, could you please? Yeah, I can try muting. I can't do it. No, I can't do it from here. Oh, no, yeah, I can do it. Oh, no, I can't unmute someone else.
Hello, sorry. Do you hear me?
Hi, morning.
Good day to you.
Yeah, morning. Okay, so yeah, as I was mentioning, so working on the resilience of plants to cope with global changes, both on using a molecular approach and using an ecophysiological approach, measuring functional trait of plants. And then yes, to integrate these 2 approaches and see and study this kind of adaptation of plants to climate change. Then we also have a huge historical collection, mainly represented by herbaria. We have, I don't remember how many millions of sheets, half a millions of herbaria sheets. And yeah, in the last year we started, and the process is in an advanced stage, process of digitalization of these herbaria sheets, so that we will get a lot of data, and we're planning to analyze this data and to produce research from this activity. And of course, we still have groups working on conservation, and the idea is to perform studies to integrate conservation across spatial and ecological scales. So, for example, we will talk later about the newly developed germplasm bank here at the Botanical Garden. Okay. So one example for, let's say, each of these pillars regarding biodiversity, as I mentioned before, last year, my colleague Francesco produced and published for the first time the chromosome level of the genome, okay, of the Goethe's palm. It was the first time for this palm. And we are using then this information in order to integrate, as I mentioned before, the molecular and the functional approach. So thanks to a collaboration with the Montgomery Botanical Center in Miami, we had access to a collection of younglings of Camerops humilis germinated from seeds collected along an elevational gradient in Morocco. Okay. And the plants were germinated. They were grown for a few years in the Botanical Garden in Miami. And last year we went there to perform molecular analysis and also functional one. And then we want to understand whether different population that differs on their functional strategies also differs in their genetic diversity. Okay, so the projector is not helping here. We have multiple population. You can see a point here, but let's say along this area here, there are multiple points. This one, you can see it here. Okay, so that we are covering a large spatial scale. Okay, so regarding the functional approach, we are measuring parameters that can inform us on the functional strategies that these populations, the plants belonging to these populations, are adopting. I won't go into the detail, okay, about the parameters and the traits, but we have traits related to the drought tolerance of these species and traits related to the leaf construction costs, okay? Here in the slide, you see 3 populations. Each dot is an individual of the population. The color represents the density of occurrence of individuals into this ordination, or we call it functional space, okay? And the position of the individuals within this ordination is determined by the value of these functional traits here, okay? So that the populations that have a higher density towards this region of the space are the ones with the higher drought tolerance, and the ones that are more clustered, more dense in this region here are the one with higher leaf construction costs, okay? And you see, for example, if we compare these population with this other, you see that the first one has a higher concentration in the upper part of the ordination, so lower construction cost, while the second one is highly concentrated in the lower part of the functional space, so higher construction costs. And right now we have genetic analysis ongoing, and then we want to understand whether the population that differs in ecological strategies also differ in their genetic structure, and which region of the genome are responsible for this separation, so that we can find correlation between genes, their functioning, and then the ecological strategies adopted by plants. Okay, so we hope to have results in soon. Okay, yeah, very soon. Okay. Then, of course, we mentioned the resilience pillar. We also have to consider, of course, that we're living in a world that is changing, and this means that the climate is changing, but also how we are using our lands is changing. And of course, this have an effect on the diversity of plant and on the ecological strategies that are more or less suitable to these changes. Okay, so as a direct consequences of all these changes, we registered an increase in the forest mortality. So this is a map showing the points in the globe where we had an increase in forest mortality in the last decades. And as the map is showing, the phenomenon is global, okay? So we have points, each dot represents the forest with a certain intensity in mortality. And you see that all around the globe, we have points where we registered an anomalous mortality of forest, okay? So that the idea right now is to understand, is to measure the functional characteristics of the species in order to understand which one is more or less suitable to these changes, and also to understand how the ecosystem, how the communities that colonize this ecosystem will change according to the changes in the climate and in the land use, okay? How to do that? By measuring the functional traits, okay, which are any kind of measurement, could be a morphological, physiological, or phenological characteristics that can be measured at the individual level, okay? So I have a trick for explaining what is a functional trait, okay? So just imagine that, think that this is a leaf, okay? So this leaf has a certain area, okay, and a certain mass, okay? So this leaf is a big leaf, okay? So it has a very big area. This means that it can intercept a lot of light, perform a lot of photosynthesis. And so I expect that the species with this kind of leaf will be highly productive, fast-growing species, okay? Then we can have species like this, okay? They invest the same biomass, okay? But actually the leaf area is lower, okay? So that the light intercepted is lower, the photosynthetic rates will be lower, and so that the productivity of the plants will then be lower. And we can go on and on and on, okay? So that we can have a range of species, okay, along this continuum, okay, of leaf shapes and morphology, okay? So, which is the advantage of having one or the other strategy, okay? So, these kind of species, they invest a lot of energy for a small leaf that is not so, let's say, efficient in making photosynthesis, okay? But actually, this leaf is really tough, okay? So it is really hard to break it. So it can withstand with, even if the climatic conditions are changing or it's not that suitable for herbivores and so on. So, for example, we do not eat laurel salads, for example, okay? The leaves are too tough, okay? While on the other hand, these kind of species, okay, they are more vulnerable, of course, to changing climatic condition, okay, but they are more efficient in making photosynthesis. So they can concentrate the metabolic activity in few weeks or in few weeks of the year and perform the vital cycle in a lower time. And then even if the conditions become stressful, can lose the leaf, and it doesn't matter for the plant, okay? So just by measuring one simple trait, two simple traits, the leaf area and the dry mass, we can already describe specific ecological strategies of the species. And the idea is that with functional traits, you can do that. Okay? According to the function that is linked to the functional trait, you can describe, okay, the ecological strategy of the species, okay? And we are lucky since there are biophysical constraints that limit the combination of different functional traits. Okay, so for example, it is impossible to have a species with this very high investment in leaf construction, so in dry biomass per unit area, and also with very large leaf. Okay, so this is a so-called trade-off. There is a trade-off between how much of biomass can be invested per unit leaf area. And these trade-offs can help us in defining the ecological strategies of the species. This is one of the most known trade-offs. This is the leaf economic spectrum. And again, I just wanted to focus on three main functional traits. Leaf mass per area, this is the ratio between dry mass and leaf area. The photosynthetic rates, and this is the leaf lifespan. So how many months a leaf lasts, okay, before drying out or falls from the tree, okay? So species with higher values of leaf mass per area are species that invest a lot of biomass for building leaves. And in fact, the photosynthetic rates are the lowest, okay? But on the other hand, the species here are the ones with the longer leaf lifespan, okay? They have leaves that last longer. And on the opposite side of this trade-off, we have species with low construction cost, high photosynthetic rates, but with leaves that last for lower time, okay? And so that we can divide species according to the so-called acquisitive conservative continuum, okay? So we have conservative species, tough leaves, low photosynthetic rates, but that last longer, compared to acquisitive species, okay? So this is just with 3 traits. We can do better, okay? And this is something that we are doing, let's say, in the last 10 years, okay? So the idea is to increase the number of traits, increase the number of functional dimensions, so that we can fully describe the ecological strategies of species. This is the global spectrum of plant form and function, a seminal paper for people working with functional traits. So here the authors used existing databases. Each dot is represented by a species. We have thousands of species for which we have measurement of 6 functional traits. And they highlighted 2 main dimensions of this functional space, okay? One is related to plant size and leaf size. So here, for example, you have leaf area, and that is the second component, second dimension. And the first one is represented by the construction costs, okay? So that, again, with these 6 functional traits, we can describe the ecological strategies, in this case, worldwide, considering the global, okay, diversity. And again, even in this functional space, even in this multidimensional space, we can highlight, okay, the acquisitive conservative continuum. Okay, why I'm telling you that? Okay, since here we just have two dimensions related to leaf size and leaf economics, but we know that there are other dimensions, other functions that are important for the plants and that might be important in this for describing their ecological strategy. So I'm really interested into plant hydraulics, and so one of the mission here at the Botanical Garden is to include this kind of traits related to water use and acquisition, drought tolerance, water transport efficiency into this global spectrum of plant form and function. Okay, and this is something that Julia is doing right now, starting with palms. Okay, and the basic idea is that we can elevate the role of botanical garden not only as conservation places, but also as laboratories, living laboratories for studying plant diversity, and in this case, plant functional diversity. So here, already in 2018, these authors here just compared the variation of the functional traits, the most non-functional traits in global data sets that are represented by dots here, with the one considering only the plants that are cultivated in botanical gardens. And as you can see, there is not a very completely full overlap, but there is a huge overlap, okay? So that, yeah, we are preserving species that represent most of the global functional diversity, but not the whole, okay? So one thing that we can do is to create networks between botanical gardens and measure the functional diversity that is conserved in the botanical gardens. And we can, for example, use this information to guide the management of the collection that we have, so that we can, for example, introduce species that can help us in expanding, okay, the functional diversity that is preserved in botanical gardens, okay? And yeah, this could be another criteria that could be useful to understand how the role, or which is the role of botanical gardens and how to expand their role globally. Okay. Then the very last point, okay, I wanted to present, let's say, the newly developed germplasm banks here in the Botanical Garden. So in the last year, we acquired few freezer, okay, minus 20 freezer that we're using for storing seeds. And the idea is that we want to improve the activity related to this facility. We are planning to hire a person working fully developed to the activities of the germplasm bank. Okay, so of course, conservation in botanical gardens is not only represented by living collections, as we mentioned before, or plants that are cultivated in the botanical gardens, but also, of course, storing seeds is a way to preserve diversity, both on a genetic and on a functional point of view. And it is not easy, of course. As I mentioned, we had to hire a person that is fully devoted to this activity, since it is not just collecting seeds and put them into the freezer. Okay, so there are protocols that must be established according to the type of seeds, type of plants, and so on. And so this is what we're doing right now. Okay, starting from the collections that we have here in the botanical garden. Okay, but the germplasm bank could be useful to understand again how to improve the activity of the botanical garden and to increase the robustness, let's say, of the conservational value of botanical garden. I'm presenting an analysis done in the last year by a member of our laboratory. So here he mainly focused on alpine flora and on alpine botanical gardens. And the idea was to understand how much of the diversity of alpine flora is preserved in alpine botanical gardens and in alpine germplasm banks. Okay, so this is the network of botanical gardens that it was able to develop. Each dot represents a botanical garden, and the size is related to the number of species that are cultivated in that specific botanical garden. Okay, so using the alpine database of alpine flora, we reconstructed the tree representing all the families that for which we have at least one species cultivated in the botanical gardens, and the size and the colors of for each family represent the number of species globally, okay, cumulatively, sorry, preserved in the Alpine Botanical Gardens, okay? And this is a similar tree. No, sorry, this is not the one conserving botanical gardens, but this is the tree related to the flora present in the Alpine region, okay? So this is the family present in the Alpine region and the number of species for each family, okay? This tree then represents the family that are actually for which we have individuals planted in the network of botanical gardens or for which we have seeds in the germplasm banks. And the red color represents families for which we don't have either a plant in the a living plant in the botanical garden or seeds in the germplasm bank, okay? So this information could be used for next activities so that we can go into the field and collect seeds, starting from seeds, collect them and store them in the germplasm banks. And for these botanical gardens, whether they have to decide which new species to introduce or new collection to introduce in the botanical garden, they can use this tree as a guide, okay, to understand which species is better for this aim, okay? And this is it for our webinar. If you have questions, we are here. Yeah, thank you for your attention.
Nice presentation.
Thank you. Thank you.
You do a lot. So it's the Renaissance. It's impressive. Congratulations.
Oh, thank you. You have to congratulate with Francesco, which is leading the labs in the last 3 years. Yeah. Yeah. Then we hope that some of the hints that we give could be helpful for you and for other botanical gardens. And also, if you have other framework for example, assessing the quality or the conservational value of the collection, would be great to have such a discussion and understanding how you do stuff, okay, and so how we can learn to improve our way to manage the collections, okay.
I think what can present our.
Yes, I have presentation.
Yeah, you can come.
It will be shorter and focus mostly on our collections. What we have to present you, because we do not have such good results of the biology of plants. So, however, we have also the branch, which is called Laboratory of Plant Communities.
Okay.
Tissue culture, but I think this details will present in Polish. And the webinar will be meeting all together.
Maybe the presentation didn't start. I don't know why. I don't remember. Oh, no. Okay. You can use the arrows or the mouse to switch slides.
Thank you very much. Hello, everyone. I'm Alexandra. I'm working in Botanical Garden at Arboretum Wojsławice. We are arrived here for Arqus Verde. And I want to introduce to you some, all of our national collections are mainly located in two places because our garden is separate for 2 places. The main garden is in Wrocław, and we are working in Arboretum Wojsławice when it's mainly place for a historical collection. Yes, yes. There is a view of the cathedral from the Botanical Garden in Wrocław, and our living collection, our long-term collection, are biology resource for us, for research, for conservation, and for education. And there is our collection. Some of this is located mainly in Wrocław, and some is located in Arboretum Wojsłowice. And we have ibis, bromeliad, anubias, sword plants, water lily, daylilies, rhododendrons, peonies, boxwood, hydrangeas, witch hazel, and periwinkles. Several collections are very important for us and for our research, for our education and conservation because of their size. There's one of our rhododendrons in Arboretum Wojsłowice. Some of specimens are over 100 years old, and yes, and they are no longer available anywhere to get. Our collection include ornamental plants and wild ones, taxa from all over the world and from Poland. Our garden support university teaching and laboratory observation. Our tissue culture laboratory allows us to provide some plants and to preserve genetic material. Arboretum Wojsłowice, when I'm working and we are working, it's famous because of its rhododendrons national collections. Yes, and hydrangeas.
It's very old in the Poland collection.
Yes, it's the oldest part in this collection. Our largest collection is daylilies, and it's granted by American Herbaceous Society by this day garden status because of their size and the taxa. And then we are coming into Echinodorus. It includes 100 species. Anubias. It includes 18 species. Our smallest collection.
It's quite interesting collection because we keep these plants in the aquariums. We have 29 aquariums with sweetwater plants, and 2 of them are national collections. What does mean? Those are the biggest on the one in our country. Usually we have more of all species which are in the north. Especially Anubias, we have all species from Africa, Echinodorus from our Aquarius. We have most of which are in South America, and a lot of cultivars which are used in aquariums, like decoration in aquariums. Each aquarium has 1,800 liters water, and are divided in geographical regions. And also we have fishes from the same regions, from the zoo, from Zoological Garden, Wrocław. So this two collections are really big. I think there are no representative other botanical gardens in Europe, at least.
And it's hard work every day. The aquarium downstairs.
A lot of fish.
Yes, true. All the aquariums are designed in different styles. We have also one greenhouse, which we have also gardens where the plants grow not under water.
And is it designed to be like a self-sustained ecosystem, or do you really need to take care of algal growth or Yeah, we have to. Yeah, they are quite fragile. The water, especially condition of water and of ice, very difficult to really to heal the algae. That's why we use osmotic water to use the house. You have to change the water in the aquarium.
Yeah.
Of course, not all 1,000 there, but a third of the home.
Okay, and then water lilies, we include only winter hardy water lilies and other ponds. And it contains 19 species.
Very interesting national collection because we have species in all of them. So we have both first national collection, but mostly we have cultivars, which are different. We have also hybrids between tropical and European species, which are a little bit renewable. And of course, a lot of different taxa with different shape and flowers. Also, because we have small legal effect in the garden. We have also the second because it's very important. The branch came 90 century throughout garden or people's established and was used as a protection community zone. After the Napoleon War, this special fort was destroyed by French people during, of course, Prussians. This fort protected the old town against enemies. So after that, the land, the empty land, and the town decided to leave us this land to the university, and in 1811 our Botanical Garden established in the role and so 200 years old and very impressive, of course, art novel style, romantic places to rest, of course, a lot of paths arranged romantic way.
Yes, and it's mainly in Wrocław, on Botanical Garden.
But the branch, the daily leaves are in the Wrocław, but also in Alberta was a bit, we have many points there. So we have the collection hours of them.
Yes. And next, bromelias. It's mainly kept in a humid greenhouses also in Botanical Garden in Wrocław. It includes over 500 taxa and especially Tillandsia, epiphytic Tillandsia.
Maybe next year we'll move this collection to another greenhouse, bigger and with better condition.
And the new one,
Yeah,
okay.
we are starting to rebuild the old 19th century greenhouses, townhouse, Australia and Victoria, the complex of 3 greenhouses from different regions. But now with the only tropical, it's easier to manage. Because they are not so large or so huge, so that's why it's the only one condition. Climatic condition we focus for the real tropical species. From Amazonia, with of course Victoria Amazonica, Victoria Regia, it was present in Wrocław since I think half of the 18th century. The Germans, of course, collected all the species. It was quite well developed botanical garden because they traveled over the border. Very interesting species because the climate in Wrocław is much milder than the surroundings. We are in a big city, a big river. That's one of the warmest places in Poland. So they introduced a lot of species from North America and some part of the world, and they were introduced first with our garden as well as in Arboretum Voice.
But maybe it plans to dwarf farm in the new greenhouse in the Wrocław.
Yes, yeah, it's possible. But we can try also planting it outside because we had in pots, but we keep it in the cold greenhouse during the winter. During the summer and spring, yeah, outside. Okay. And grows like
Then easy. National collection includes all the heteros species, all the wild species, and one botanical form. And there is 500 cultivars.
Very interesting and different leaves, color and
Yes, the shape, color,
shape,
and
yes,
yeah, actually,
for me it's interesting.
it's quite interesting collection. That's right. We have all species. Depends on the taxonomy, 16 or 17. We have all, of course, always there are not some doubts about the identification. That's I think it's not unusual gardens, but the varietas of Federa Felis, it's really crazy. It's really crazy. And quite interesting about the genetics. What caused such change in because most of them were found in the native species as a result of some genetic change itself.
We have a Polish cultivar, Federa Felis.
Yeah.
peonies. Peonies mainly houses by in Arboretum Voice because there's a really large space in the garden. We have 32 hectares for our collection, so there's a space for.
Wow.
Buxwood, it takes 150 taxa. And an engagement plant species. Yes. See this species?
Of course, we have a lot of species because the taxonomy is almost it's all on the list, so it's not difficult to learn number of this group.
Yes, and we believe that when we are keeping them in the Botanical Garden, we support the legal trade of them. And plant protected species, plant species in Poland. We want to educate our local community about our native flora, and that's why we caring about plant protected in Poland. Thank you very much.